Talking about a collaborative program of research with Rufen Vogels and Macaque there in Belgium, and we've been studying object representation in the Macaque inferior temporal cortex. Obviously, the experiments, are done in collaboration with them. And the inferior temporal cortex shown here is since the work of Charlie Gross in the late 60s. Is an area generally believed to serve object recognition. Tube responding to the cells occurs between 80 and 100 milliseconds after an image is painted on the retina. Well, in a quarter of a century or so, since Charlie Gross first discovered that these cells were driven by visual images, the dominant methodology was that of a beauty pageant from a very small town. So an experimenter would take 8 or 10 images and show it to the monkey, and you'd see which image fired that cell the most, and that was the beauty pageant winner. And the selection of stimulus was quite haphazard, and it almost led to, well, people were skeptical, to a belief of a grand object cells. But more recently, some work in Katie Tanaka's lab in Ricken, and specifically some work by Macaque Tanafuji, has been a bit more systematic. And what Tanafuji does is optical imaging of this region, and it takes a complex object and shows it to the monkey. And you find that you get spots in this region of Itti. And then Tanafuji goes down and records perpendicular to these spots on the surface using a method pioneered by Tanaka in the sense of you reduce the objects and you start with a complex object and you take off parts to see or try to assess what the cell might be responding to. So if you take off the barrel of the fire extinguisher, you find the cell still firing. You take off the hose, it's still firing. It doesn't fire to the hose alone, or so it does fire to a hand, or let's say a cat's head, indicating that maybe something spiky is driving this cell. In another case, again, this is another spot. You again reduce it by removing the barrel. The cell still fires, but now when you show the handle, the spiky handle, the cell doesn't fire. It does fire to, let's say, the hose very well, but not to a straightened segment about that orientation. And in general, cells at each spot seem to respond to a simple shape or two, or characteristics of a simple shape. Now, the problem here is that this methodology, both in the selection of stimuli and the manipulations that are made, is pretty subjective. You don't quite know if you would have tested the cell in different ways, what the cell might be responding to. So this seems to be a general conclusion that it's rare that you need a whole object, if it's a complex object, to drive the cells effectively. In this region, there's enough evidence to indicate that many, or not most of the cells, are decidedly nonlinear. So simple reverse correlation methods where you just show pixel noise to the cell don't seem to give you very much. At least certainly Rufen Vogels has tried that, but it's not getting anything that way. So what I'm trying to method is to be guided by a theory. And the one we selected, perhaps not by chance, is Geon theory, where you assume that objects, for purposes of recognition, are represented as an arrangement of simple shape, simple parts called geons. And these geons are distinguished by non-accidental properties and viewpoint invariant properties along with generalized cone dimensions. And I'll mention that in a minute. But one, for example, is whether the axes of a part is straight or a curve, or whether the cross-section as it's moved along an axis remains constant in size, as with the cylinder view of the brick here, or in fact, let's say expands and contract and so on. And that this information can largely be determined from edges produced by orientation and depth discontinuities. Now, we're wrapped to a generalized cone representation, which was formulated not by, proposed originally not by Laurent, but by Thomas Binford, is that it's not appropriate for highly complex irregular shapes, such as bushes. Or just, in fact, you could accuse it of being a theory of all the objects that God didn't make. That's not quite true, because you can actually do a fairly good job for distinguishing animals that people readily distinguish. But the question, if one's interested in this as a possible model of human or primate vision, then indeed one might ask the question, are people very good at distinguishing amongst complex irregular shapes, such as different bushes? Or are cells in IT sensitive to those differences? So we'll look at here's just, for those who might be familiar with this notion of a non-accidental change. Let's say you have a shape like this, and you straighten the axis. That would be apparent from most orientations in depth, other than, let's say, an accident. But you can vary a metric change as increase the curvature of the axis. And you can make these differences, and in all the experiments I'll be describing, these differences are scaled either in terms of pixel differences or from a wavelet-type model, or the V1-type model proposed by Christoph von der Malsberg, it's the Mundhenk model. So one can scale differences, let's say a metric change versus a non-accidental change. And in fact, it's impossible to do this work in any principled way without such scaling. And here's just, for those who might be unfamiliar with generalized cylinders or generalized cones, it's just ways which just a formalism for describing shapes. And you can do it with 2D shapes as well, in which case the cross-section is just a line. And I'll be talking about some experiments with just 2D shapes. Well, first let's look at the sensitivity of single IT cells to non-accidental versus metric properties, as well as whether the tuning of these cells might be along generalized cone dimensions. For example, does a given cell respond to a curved axis, and it doesn't care if it's a curved cylinder or a curved brick or a curved wedge or a curved cone? And is that tuning may be independent, again, of other possible variations? So we're moving, what I think was to take a set of stimuli that would be very systematic. I know these look a little strange. We hunted for responsive cells with two-part stimuli, and then we tested them, if possible, so they remained responded with just a single part. And half the time we were able to get good responses with just a single part. And the pictures I'll show will be from these single-part stimuli, though the results are the same in terms of tuning when you have a two-part object. So this is that comparison I mentioned before. You can have, if you start with the standard stimulus, and this is why they look a little strange, you could, let's say, vary it so you make a metric change. For example, you could have a slightly rotund barrel and make it much fatter, or you could flip it down and have the sides parallel. Or the axes could be slightly curved here. You could make it still more curved or straight. And again, these differences, metric and non-accidental, are scaled both in terms of pixel and wavelet type measures. And this first experiment will be in terms of pixel type measures. So, are the cells more sensitive, in a sense, of modulate more to differences in non-accidental properties than metric properties? And here are four lovely cells, and in the sense that they do. So we're interested in, again, modulation up or down. So let's say if we start with a slightly curved pyramid, we curve it a bit more, we get maybe a slight change in firing, but much less if we straighten the axis. And these differences often are quite subtle, so we get a fair amount of modulation when we cross over the non-accidental border. These non-accidental properties can be demonstrated to be quite critical for easy recognition when objects are shown at a novel orientation and depth. So this gives us this maybe the basis for object constancy, or the ability to recognize objects in new orientation. Here's a case where, again, that slightly rotund file is still fatter. There was very little change of firing, but when we straighten the size, that is the cross-section at constant size, we get a great increase. And you can see in all cases the non-accidental property change in these examples, led to more modulation than the metric change. And these differences are scaled according to pixel differences. And here are just results. So given the scaling, we can now compare, and these are results from two monkeys, Yelko and Rambo. And here is the percent modulation. This is for, and the monkeys gave quite similar patterns. Quite nice. So here is the modulation. Given the standard, we set the magnitude of the non-accidental change in terms of the physical stimulus change to get this much modulation. If we have an equivalent metric change, we're over here. This is MP2 is just a level. It's a slightly more or a larger stimulus change than the non-accidental change. And you see there's less modulation. In general, you get about 50% more modulation with a non-accidental than a metric change. This was really significant in both monkeys. There's another effect, which I won't describe, but we can discuss later, that when you change a metric property, we change an image similarity by changing a metric property or rotating the object in depth. And when you do that to equal extents in terms of stimulus change, you get equal amounts of modulation, both of which are less, considerably less than the non-accidental, than the modulation you get with the non-accidental change. This indicates that the effects that someone might see from rotation and depth might simply be that of physical similarity. There's no counter, maybe, for orientation itself. Now, are these cells responding to individual dimensions of, let's say, generalized cylinders? And here's an experiment that we recently completed where you start with two basic shapes, a triangle and a square. You can vary the axis curvature of this, or the positive curvature of size, the degree of that, or the metric variation, or the positive curvature of the square, negative curvature of both. And we had another dimension of just tapering over here. And in general, red is indicating more activity of the cell, bluish or darker colors less. And you can see that this particular neuron responded very strongly to negative curvature. But it didn't care if there, and this other dimension, so going down this way is negative curvature, going across here is tapering, or a change in the parallelism of the sides. And the sensitivity to the negative curvature was quite independent, and this is generally true of, let's say, the tapering. And if one does then, for all the cells, say, multi-dimensional, here's another example where this cell was sensitive to the, let's say, curvature of the axes, like the straight axis, and like less curved axes, and as you curve the axis more and more, less so. But the sensitivity to this straight axis was independent of the aspect ratio, or the length of the object. And if you do a multi-dimensional scaling of pairs of these variables, you find that the cells very nicely, this is a bunch of cells, in this case 98 neurons, very nicely distinguish one attribute, in this case, curvature of the axis, from tapering, or negative curvature of the sides, from positive curvature of the sides. They are surprisingly well behaved this way. Let's look at another issue, which is a discrimination among regular versus among irregular shapes. That is, if you have, let's say, a round table and a square table, you may readily notice that difference, or a topiary, where you make a spherical bush versus a square bush. You know, you have the geontopias or something like that, and versus this regular wild irregular bushes. But let's say the differences are equal according to pixel differences, or V1. What about the cells? So one can ask two questions here: your discrimination among regular shapes or irregular shapes, as well as the discrimination of a group of irregular shapes from regular shapes. So even though we might have difficulty distinguishing among the irregular shapes, we know they're irregular. And you can show that psychophysically in experiments. Trust me on this experiment. So here's the testing of these cells, and we use the group of shapes. So here's a regular group, and it's maybe surprising, but within groups, there is, the differences are equal according to pixel differences. Even though you may be much more sensitive to these differences, but you're using your IT. So here we have a complex. Here are relatively simple shapes. These are round and these are square. We can ask, what about those differences? And let me just give a summary of something today in a moment. That people, or the cells, also people, we also ran a sorting task, that people are quite sensitive to the regular differences and quite insensitive to the differences in these irregular shapes. Within groups, even though the differences, again, according to metric changes, are equal. And this was what the cells in IT did. Here is the modulation, and within the regular group, there was more modulation, less so within the irregular groups. And here's where typical cells might be firing within this set. So here's a cell that likes rounded, maybe rounded compact stimuli, some regular ones here. Here's a cell that likes, let's say, highly complex stimuli. Here's a cell that has a strong orientation preference. And what we can do is then multidimensional scaling for pairs of these groups here for these variables, regular from irregular, complex from simple, curved versus straight. And in general, IT cells, not in general, IT cells and people, just to give you the take-home lesson here, because there's some data in a moment, make these distinctions quite well. Image-based models, either pixels or the Bourget, or a model proposed by Riesenhuber and Poggio, this HMAX model, don't distinguish those differences. So here's just an example of the regular from irregular complex, and here is multi-dimensional scaling of the activity. And the white dots are irregular stimuli, complex stimuli. The black symbols are regular ones, and you see that they're nicely distinguished. The second dimension here is orientation, which is that this group here with the strong orientation is distinguished from the others. And one can do this with all the variables. Here is something, curve versus straight or regular stimuli, relatively simple. And again, the multidimensional scaling distinguishes that very well. That's the black figures from the white. Here's some sorting behavior in humans, well distinguished on that. But if we take pixel energy differences, we don't see any nice separation. And here's the Mausberg or Bourget model. Again, you see black and white dots intermixed. And over here, the HMAX model, again, we don't see a nice separation between the regular curve and regular straight stimuli. Let me go through this quickly. What about edge using edge information? In general, this is an experiment which I published recently. If you vary the direction of illumination, you can get large changes in terms of V1 similarity, that's a wavelength similarity. But then you can look at, this is like illumination from front, left, right. But then if you look at IT cell tuning, for the most part, the cells seem indifferent to the direction of illumination, as are people in most experiments. Also, the tuning, the sensitivity, the metric, greater sensitivity to non-accidental versus metric differences, it's largely maintained when we take silhouettes or even a wide range of the stimulus. So, in summary, consistent with this simple component or genome account for human performance, cells and object areas of IT respond well to one or two part objects, are well tuned to the dimensions of generalized tones, maintain the selectivity of the tuning that was established with original shaded images when presented with images preserving only depth discontinuities, and show greater modulation, which is greater sensitivity, to non-accidental and metric differences or differences among irregular, simple, or complex shapes. And they strongly distinguish on a population level simple from complex shapes, regular from irregular shapes, and groups of shapes different in their non-accidental properties. These effects are not accounted for by free image-based models. Thank you very much.
Since most of the distinctions that these cells are making junction in the image. When you have two curves coming into a point, or etc. And kind of in the spirit of the simplifications that Macaque tries to sort of stripping away, one wonders whether these cell bursts are essentially responsive, they have a favorite junction type. And if that would be the case, that's a little bit surprising in the sense that it's a pretty simple measurement actually to be making. And for example, if you look at, I guess Ed Connor's group, they do lots of cell stimulation before they find almost even more complicated selectivity there. This is one stage further. So could you comment on that?
Well, okay, so Connor finds, for example, cells that are L-vertex detectors. So you have two contours co-terminating at a point. These are actually, this is something, actually, what stimulated that experiment was that model that Hummel and I had here. But even earlier, there's this work by Biederman showing how critically important L vertices are for defining the ends of the surfaces. Well, a feature like that are in, for example, our complex irregular stimuli as well as the regular ones. So it doesn't distinguish that. More importantly, there's another experiment we did in another collaboration in Szeged, Hungary. We took complementary images. This is an experiment I did some years ago, where you delete from each geon, you delete half the vertices and contours. And if you look at them side by side, they look identical, but there's nothing identical. So each image has half the contours, half the pixels. You superimpose them, you get an intact image without any overlap in contour. Well, the cells in Itti seem to respond the same way. That is, they seem to be responding to the parts that are and insensitive to the specific contours and vertices, just as people are. So you do use those stimuli in what are called priming experiments or matching experiments, and it actually takes scrutiny to realize there's anything different, even though in terms of any type of image measure, they're dramatically different. So indeed, we're using that type of tuning to generate the geon that Connor did. We didn't vary that in this experiment systematically, but it would be hard to imagine, since those L vertices are distributed through, let's say, our complex and irregular stimuli, that would be the defining.
This way, there are a lot of ways of attaching a part to a whole. And looking at everything we're looking at, sort of imagining a case where you're cell-specific to certain part, cell-specific to the thing to which it's attached. But do you see any evidence that there's anything responding that cares about how these things are attached?
That's a great question, and actually is the critical one, I think. Vision about the I think is a big news space. All vision should be view-based, other than, unless you're arguing about ESP. So, but the question is, what really distinguishes the models, whether you have explicit relations. And we found it difficult. We tried thinking it through, and there's an experiment that Ken Haywood presented at BSS, which is the first study that I know, and this is using fMRI, and it was run at Caltech, where you're varying the relations amongst parts as distinct from the parts themselves. And they have, it turns out, quite distinct neural signatures. So, but that's the first study. There's still a lot more control experiments that need to be done. But that's really I think, the fundamental difference between these classes of theory.
It seems that although the neurons can't distinguish between the direction of light, the color of the objects, the people can discriminate them very well. So I just wonder how you think the information about shading is combined with the information about object shape, because at some point they have to come together.
Given enough time, people can do almost anything to get, they figure out the cube root of the moment of the stimuli. When you do matching experiments, there's no question, you look at the stimuli and you can figure out which way the direction of light is coming. Marissa Niederhauser, some years ago, did an experiment where she did object matching experiments, both familiar and unfamiliar objects. And the matching was quite independent, and she varied the direction of illumination. So that's in the same trial, sometimes the direction was the same, and sometimes it was different, and there was no effect of that. Now, subjects could look at the stimuli and figure out, yeah, look, this surface is darker in this one than that one, but it seems in this hot object matching case, where you're just trying to say, these two are the same shape, that you're largely independent of that, though there can be some extreme cases where you do see an effect. But you're surprisingly insensitive to the direction of illumination for these objects.
Well, the reason I raise it is people sort of act as though IT is the end, where the object is recognized and so on. And it seems that there are other properties that have to be related to the object, and wherever that they're related is going to be closer to the end than IT, which is apparently according to this new shape.
Well, it's not only, I'm maybe being too strong, but you're quite right. So we can get in that experiment with Rufin, where we vary direction of illumination, you get some effects. But what's interesting is what you see, the dominant effects are what people seem to be doing very readily, very quickly. And then you wonder if you need some top-down process, maybe a front area doing research to come up with some of the other properties when your real goal is to try to just recognize that these two objects are the same or not.
Let's time for the speakers.